video is brought to you by Vita Bee Health. Thanks Vita for sponsoring this channel. Please check them out, link in the description of this video. Hello everyone, in today's episode I had the pleasure to speak with Dr. Kaira Wagoner, a honeybee researcher at the University of North Carolina in Greensboro. She is also the founder and CEO of Optera, the company that developed the UBeeO assay to screen honeybee colonies with high hygienic behavior. In this interview, recorded live with members of my academy, we discuss different types of hygienic behaviors and their variables, which is important when selecting queens in your operation. We also talked about how beekeepers are using the UBeeO assay in ways not imagined before by the founder herself. If you're considering to use UBeeO in your operation, Dr. Wagoner is giving 15% off on your first purchase for InsideTheHive.tv fans. Use the coupon code InsideTheHiveTV to get your UBeeO test today. Link in the description of this video. And now, I present to you Dr. Kaira Wagoner. Welcome back, Kaira, to the show. We are live.
Thanks so much. Great to be back on Inside the Hive TV.
Kaira, so I brought you here today because in my social media comments and after our conversation, apparently we're getting similar kind of questions, and there are a lot of people out there with some misunderstandings about hygienic behavior and their own thoughts based on not facts, I would say. So I think that was the idea here today, so we can both of us discuss the subject a little bit. You give your thoughts. Since you're doing this for a living, I said I know the right person, to bring here because I'm not a specialist. And I think you are the number one specialist about hygienic behavior today in the world. So.
I don't know about that. But the deeper I dive in, the more questions I have. It's like, when you study something so intensely, you feel almost like you know less and less.
But I think that's a good sign. I like to talk with my kids. When you think you're right about something, you're very likely wrong. You always need to be open-minded. Okay, this is the information I have right now. This is my position right now. But I can flip if somebody convinces me otherwise.
Exactly. Always open to new data.
Yeah.
Exactly. Yes.
I'm right. Oh, yeah. Then I just look and say, yeah, very likely you're wrong. But let's keep moving. Kaira, let's start with the following. What is hygienic behavior? There is differences, so it's a very difficult thing to define. Do you agree with me?
Yeah, and I think different people define it in different ways, but I like to define it as the detection and uncapping removal of unhealthy brood. I think it gets confused a lot of times with necrophoresis, the removal of dead brood. But I really think that it should be focused on removal of unhealthy brood. There's a different term for removal of dead brood, and so I think it's a different thing.
Yeah, I think we should focus on this too. But I see a lot of people defining little behaviors they see inside the hive, like the ability of some bees to identify mites, for example, and bite their legs, the kind of thing. Some people call that hygienic behavior as well. There are other behaviors to open the brood, but let the brood open, and they call that hygienic behavior as well. It could be some sort of hygienic activity occurring there, but it's when we were talking about UBeeO and the research that you do, and when we're trying to define hygienic behavior, I think we were focusing on the removal of unhealthy bees, and there is other people that go through a little further, the removal of dead bees. So how you navigate this ocean?
Yeah, well, the problem with hygiene is it's really complex. So I think there's at least two different steps within hygiene. And so you can have, first you have to detect the problem. So you have the uncapping behavior, I would say, is the first critical stage of hygienic behavior. And then Marla Spivak has actually shown that there are different bees with different sensitivities that do that first step of uncapping, the detection and uncapping, and then different bees that come along and do essentially make a decision about what to do next, either recap because there's actually not a problem, or they begin to remove. And so I think part of the confusion is hygienic behavior is so complex, and there's these different stages to it. There's decisions being made. And the mite biting that you mentioned, I think, is some people confuse that, but that's actually grooming behavior. So if the mite on the adult, in the what some people call the phoretic stage, or infesting the adult, then any removal of that mite is grooming behavior rather than hygiene. Also a really important behavior, and something I'm interested in learning more about. I actually just got my first grooming queens in my apiary, so I have hopefully some more research coming on grooming soon, but definitely different than hygienic behavior.
Interesting. Since you mentioned, is the same bees that uncap and remove, or are one set of bees that uncap and another set of bees that remove? Do you know that, Kaira?
I believe it's a different set. That's so from what I remember of Marla Spivak's paper, the bees that perform that uncapping are more sensitive in terms of their ability to smell than the ones that perform the removal. And that makes sense because they have to go through a wax cap and detect the problem, and they can then make even a small hole, like a pen-sized hole. Anything an antenna can get through, right? And then once that kind of threshold has been passed, then it's really easy for a less sensitive bee to come along and stick their antenna into the hole, even touch the brood directly and pick up smells that way, and then determine what to do next.
Yeah, that's quite interesting. Maybe we need to record that. They're going to do some annotations to count what bee is open and which bee is removed. I'm curious about that now. Kaira, do you have a little presentation for us, I believe, so that we can go over and differentiate those behaviors in a more visual way?
Sure. Yeah, I can go through some slides with you if you want, and then if we have questions, we can go through those, and hopefully it sparks some kind of discussion.
Yes, I have a question right now. Kyle is asking in the chat, is the UBeeO test available in New Zealand? Maybe we can just answer this one directly to him, since he's here with us.
I'm hoping that it will be very soon. So I actually scheduled, or I'm in the process of scheduling a call. I sent an email about an hour and a half ago to a potential New Zealand, Australia distributor. So we have a meeting set up, hopefully for next week, to begin the discussions. So right now I'm running projects in Australia, but it's not commercially available. So we basically have a research agreement where I can send over research materials. And I could do that on if it's a large enough scale, I can do that for New Zealand as well. But it's not yet commercially available.
Yo, interesting. Before we jump into the presentation, then let's talk about the company. How things are going? You guys are doing good?
Yeah, the company's doing really well. I think it's always kind of a trade-off between having a new technology, and you don't want to launch it too early where it's still like an experiment, and you don't want to hold on to it forever and keep it locked away in the laboratory in the ivory towers. And so, I think we hit the sweet spot where we know enough about it to know that we can identify varroa-resistant colonies. We can identify colonies that have better disease and virus resistance using UBeeO, but we also still have a ton of questions about UBeeO and how it works. And so I'm just really glad to have it out there and have beekeepers now. I think we've hit 8 countries now that have tested UBeeO in the last several years. And so we're getting lots of feedback from different groups. I'm seeing amazing ways that beekeepers are using UBeeO that I hadn't even thought of myself. So I'm just really glad to have the technology out there for those that are interested to explore, and excited to see what comes with it.
That's something that I experienced in the past. We put the product in the market, and then all of a sudden the people are using it in a way that you just, why I didn't think this before? Or can you share some of those that people are using a different way?
I have some pictures in the presentation.
Okay, so let's go through the presentation then. I'm curious to see what we got here.
Can you
all
see
right. Yes, we can see the slides now.
All right. So I just thought I would discuss a little bit today about something that's been on my mind, which is that when you use different hygiene selection tools, different methods, you get different outcomes. And it seems like a pretty obvious statement, I think, when I read it and when I think about it, but I think it often gets overlooked. And so I kind of just wanted to discuss a little bit about the different techniques. I did have just a reminder here about what hygiene is: the ability to detect, uncap, and remove unhealthy food. So this video shows some hygienic behavior. This is from a UBeeO assay. You can see some uncapping, a brood being pulled out, a pupa being pulled out there for removal. And I just scanned down to show, it's not the whole frame. It's just that section that's being uncapped. And I just really do think that, generalizing hygiene, that it's a really promising and sustainable option for controlling pests and diseases in honeybee colonies. So I think it's the future. There's just the caveat that not all hygiene is created equal. And I think that's especially true when it comes to Varroa resistance. So past studies have shown us that with hygiene, you can get pretty good resistance to things like chalk brood, American foul brood, but that's not always the case with the Varroa mite. So I just listed a couple studies that kind of showed different results with respect to Varroa, but I think the main reason we get these inconsistencies when we look at the effects of hygiene on Varroa resistance, or the relationship between the two, is that there are so many different methods for measuring and selecting for hygienic behavior. So this chart just shows a few of the pros and cons of different methods. So I have here listed I think the main selection methods: UBeeO, Harbo, also called SMR, the pin-killed test, the freeze-killed test, and genetic analysis. And then I kind of an out group, I put grooming on there because it's another stock selection method for resistance to Varroa, but it's not specifically hygienic behavior. And so I think the main point here is that really, as much as I'd like there to be, no method is perfect. There's no silver bullet. There are practical trade-offs for each of these different methods. So folks just have to figure out, what works for them in their operation. I think two important points with Varroa resistance, the selection methods that are best at getting that really accurate Varroa resistance, we tend to see that they come with a cost, whether it's a financial cost or a higher labor cost. They just aren't as necessarily as inexpensive to perform. But on the flip side, when we have those easier, less expensive methods, we tend to get less Varroa resistance. So it's a question of, how important is Varroa resistance to you in your hygienic selection. And one thing that I think is important to note too is these two methods, pin-killed and freeze-killed, that are slightly less effective or less effective against Varroa, involve not necessarily hygiene in the selection process, but necrophoresis. So necrophoresis is the removal of dead individuals. We talked about it a few minutes ago. But a study came out in 2018 that showed, there are these death pheromones like oleic acid that trigger removal of dead brood. So this study was done with freeze-kill brood. And that makes sense that dead brood signals are different than diseased brood signals. So you can imagine that the smell coming out of a cell that contains a dead brood is probably stronger, easier to detect than the smell that's coming out of a varroa infested but living or virus infected but living brood cell. That would probably be a more subtle smell. And so with hygiene, we see there are certain compounds that are elevated in varroa parasitized and virus infected brood. And really notably, these compounds are hydrocarbons, and they're different. They're actually a different structure, different type of compound than those that are released by dead brood, those necromones. And we've shown in multiple studies that these compounds can be detected by nurse bees, and they can trigger hygienic behavior. Not to confuse things, but I'm hoping this clarifies rather than confuses. But I just want to be clear that both necrophoresis and hygiene-based selection methods can actually lead to more hygienic bees. So just because you're using freeze-kill brood, which uses necrophoresis, doesn't mean it can't be used to select more hygienic bees. You're not using necessarily hygienic response in the definition I've given here. In the selection process, you're using necrophoresis because you're removing dead bees. But the selected bees that result can be actually more hygienic as well as better at necrophoresis. So I just want to clarify that. But, despite this, they're not necessarily the same. And we've done some studies to kind of compare these two types of hygiene selection methods. So this graph is from a study that we did in over 70 different colonies that were tested for both UBeeO and freeze-kill brood. And what we found is that the responses of the colonies to these two assays are positively correlated. So that means if they score high on one, they're more likely to score high on the other. If they score low on one, they're more likely to score low on the other. But you can see that that's not always the case. So sometimes they score high on one test and low on the other. And we still don't understand all the nuances of why that is. But what we did see when we took those same colonies, that same data, and we kind of broke it down into these 4 categories of possible responses, what we saw is that the colonies that scored high on UBeeO had the lowest varroa mites, regardless of how they scored on the freeze-kill brood assay. So, what I want to say is they're similar, there's overlap, they're positively correlated, but the two assays are not the same. You're not going to get the same result if you're using one as you will from the other in terms of selection.
Kaira,
yes.
Can you go back to that slide? I just want to
absolutely
make sure if I understood very well the message here. So sometimes you're saying that sometimes, based on this graph, what I'm seeing is sometimes you select for using the death hormone, and and then when you have hygienic behavior focusing on bees more sensitive to find dead bees, basically, and these sometimes help to put varroa down. But what I'm seeing here that sometimes that's not the case. Do you have any explanation for that? Do you understand mechanistically what?
Yeah. So I think it's related to the strength of that signal that's coming out of the cell. So if you're selecting for bees that are able to detect and remove dead brood, you're still selecting for their ability to determine there's a problem and remove that bee, right?
Yes.
The smell, the strength of the signal is likely stronger. So you're not selecting with as much sensitivity, right? Because you're selecting for colonies that can detect an easier to detect signal. Whereas when you use something that's more subtle, like the pheromones associated with unhealthy brood, it's a harder test to pass, I want to say. And so if the colonies can detect that more subtle smell, that more subtle signal, then they're going to, in a real life scenario, be able to detect also a more subtle signal. Does that
makes sense? And I think that makes sense from the field when people are saying, if I remember correctly. So when you select based on the death pheromone, so you're selecting a much more powerful smell, meaning very likely only is going to work with high infestation levels of varroa mites when it's, I'm going to put this, when the shit hit the fan, actually, basically. So it it might help and you're going to see results, but might be too late. Am I being...
Or that could be the case. It could also be the case that if you have less sensitive bees, maybe they're detecting only when there's a double infestation, so 2 varroa in 1 cell, or then maybe the varroa is very heavily infected with viruses, so really does a lot of damage to the brood. And so they're able to detect those cells, but maybe not the cell where there's, still harmful deformed wing virus, but not as much of it, or maybe just a singly infested cell. Yeah. So,
if it's the case, which is apparently it is, so I don't think that's good because it's helping the varroa at the end of the day.
Well, it's better than nothing.
I know, but it's,
Yeah, so we saw, that with freeze-killed brood, the colonies that scored high, when we left them untreated, had an average of about 3.2% infestation in August. So compared to the low-scoring freeze-killed brood colonies had over 6% infestation. But with UBeeO, we got that number. The colonies that scored high on UBeeO had infestation levels under 2. And so I think you're just kind of better able to tease apart the colonies that are really able to do a better job at detecting more of those infested cells.
Cool. All right. Thank you. Now we can move.
Yeah, well, and so that kind of compares, the necrophoresis selection to the hygiene selection, but there's also multiple types of hygiene selection, right? So UBeeO and HYGIENIC are both essentially related to uncapping and removal of those unhealthy but living brood cells. And so we also ran some comparisons with UBeeO and HYGIENIC, and again we find that they're positively correlated.
So just 1 second, for people at home that doesn't know what HYGIENIC is, I think people know from my channel that what UBeeO is, but I don't think everybody knows what HYGIENIC is.
Okay. Yes. So HYGIENIC, also called SMR or VSH, which I don't like the VSH term as much, but it's essentially when you go through and you look at usually you have to do this in the mid to late summer. You go through and look at a frame, and you're looking at opening cells and looking for mite reproductive success. So that's the test I mentioned earlier. That's actually very effective at identifying mite-resistant colonies, but it's pretty labor-intensive because you really have to go through and open up a good number of brood cells, and you're looking often with a microscope, if you have one, at reproductive success of the mite in the brood cell. And so, these two are also positively correlated. So, actually high BSH, high Harbo apiaries have scored the highest of any apiaries I've tested on UBeeO, but it's still not the same. So there's a lot of overlap, but getting a high score on one does not necessarily mean you'll get a high score on the other. And we don't know exactly why that is. I think it may be related to the fact that Harbo leaves open room for both a hygienic effect and a brood signaling effect, or a brood, some kind of brood effect on mite reproduction, because both of those would contribute to kind of the test results, whereas UBeeO is focused exclusively on the adult sensitivity, so the adult's ability to detect and uncap that cell.
Thank you very much.
Sure. Yeah, I hope that makes sense.
It does.
And kind of, so I'm trying to start broad from like comparing necrophoresis and hygiene, and then come into hygiene comparisons. And now I wanted to show, like, even with the same tool, using the same tool, we can probably get variation in selection just by using the same tool differently. And so this is one of those things that I've kind of learned since launching UBeeO, is that beekeepers are using this tool, but in different ways. And so I've actually had beekeepers who say, I want to see complete removal within 2 hours. I want to see the caps removed. I want to see the bees pulled out. I want to see them gone. Sometimes there's partial removal of cells. I have beekeepers that say, you know what, there shouldn't be anything wrong with the brood that's been treated with UBeeO, because the UBeeO just goes on the cell cap. So I don't want to see removal. I want to see uncapping, but I want them to recap those cells after the 2-hour test. I'll count it, and then the bee should actually recap those cells. And so some people are selecting for uncapping, but they don't want removal. And I've actually even had beekeepers say, you know what, I don't want a lot of hygiene in my colony. I want there to be hygiene. I want them to recognize if there's an issue, but I don't want it to be like an overpowering trait. I just want partial uncapping. So it just kind of blows my mind that, one tool can be used in so many different ways. And it's really fun to see that happening, because I didn't necessarily expect that. And I'm really excited to see how the different selection programs that are kind of using these different methodologies with UBeeO, like how they kind of split ways and how they differ in terms of the bees that they end up selecting for. So that's really exciting, and will be fun to follow.
That's quite interesting. Can we go over in the hypotheticals here, you and me? Because I'm curious now. For example, what are we selecting when we're going complete removal? We're selecting bees able to smell the thing and remove things, even if it's not sick, or maybe it was a coincidence and it was sick and they removed. How the beekeeper can be sure this is a good thing?
Yeah, I don't think that there's a very high likelihood that they're all sick, in that small test area. So, I mean, the question remains. I don't think I have a clear answer. You could even be selecting for maybe a thinner cap, right? Where, who knows, maybe some of the UBeeO is going on and getting onto the brood and causing removal. I think you could have potentially genetics that kind of determine or result in bees that once a bee is uncapped, they remove it regardless. You could just say potentially it's those same bees that are uncapping in these complete removal scenarios that say, you know what, I'm going to go ahead and remove this because I think there's a problem and I'm not going to wait around. We don't really know what's happening, but I think it is definitely a more intensive form of hygiene when you get this complete removal.
That that's cool. And what we are selecting any other scenarios with the partial removal, complete uncapping,
Yeah.
partial uncapping.
With partial removal, I think it's really the same as complete removal, but they're slower. Um, they're just not as fast at performing the removal behavior. Potentially, that could mean, it's a different bee like we discussed earlier, and so it takes a little bit longer, whereas complete removal is the same bee. We really don't know, but my guess there, those two, that it's probably just about speed. Maybe how many bees in the colony are performing hygiene. So, with the multiple matings of honeybees, it could be that partial removal means that she mated with 2 hygienic drones. And this complete removal means that she mated with 20 hygienic drones. And so there's just more hygienic bees in the colony on the left than on the right. So, that in that case, you probably would want to go for complete removal if that's your breeder queen, because you're going to have more of her eggs that are produced are going to have a greater percentage of hygienic genes. With the complete uncapping, I think that's probably where I'd like to go if I was breeding bees. I'm not a breeder myself, but I just think that kind of gets you over the threshold that makes or breaks hygiene. So if you can have a cell that's uncapped, it's really easy to figure out what's going on inside. So I think the hard part of hygiene is like finding that problem and revealing it to the bees that are less sensitive in terms of their olfactory abilities. And then partial uncapping, I think, would just be kind of a lesser degree of that complete uncapping. So again, maybe it has to do with the genetics, the mating in the hive, or maybe it's just a slightly lower sensitivity to those smells.
I was talking to Dr. Stephen Cook from the USDA here in Beltsville yesterday, and he was mentioning to me that a similar scenario that we're seeing here on the screen, the complete uncapping, these might be a mechanisms to stop varroa replications. And there is some results coming out that only the little opening can disrupt potentially can disrupt the replication of varroa somehow, which is great. If varroa is making somebody sick over there and the bees just open that, don't even need to remove.
Right.
Just stop the reproduction of varroa. If that start to be confirmed in other places with other researchers independently, I think this is going to be great.
Yeah, I've seen some of those results, and I agree completely. There's been some evidence that just opening them, whether it's like the temperature or the humidity inside, can actually cause the mites to go non-reproductive inside the cell. It could be even the males that are leaving. Who knows what's causing it? But I have seen some data on that, and so perhaps that's the way to go. You don't actually even have to sacrifice the bees inside that have been parasitized by varroa, but you can actually just kind of interrupt that reproduction process. I guess the question would be, if the varroa has already started the reproductive process at that point, how much virus have they already passed to those brood? So, you might get less control of the virus if you're not removing the varroa that have been infested. But, great question to follow up on with some research.
Yeah, what I love about all this conversation, Kaira, is that I think people at home can see how flexible this whole system can be. How many things we can do, or the beekeeper with their own experience, with their own observations at their own properties, with their own bees, they can try different things along the year and see what worked best for them. And I think that's great.
Yeah, and I think, every apiary is going to have different goals, different priorities. And so it really is about beekeepers figuring out which system works for them best, what their ultimate goal is maybe, and then working backwards from that.
Yep. Fantastic. Thanks for sharing that. Let's see what we have more.
So I think just my main point here is that different methods can lead to different outcomes. And so because of that, one thing I think we need to do is be careful when we tend to just refer to hygiene as like a general thing. And so I think we need to be careful about that, because we may actually have a different result with different measurement tools. And so one example of that, and I know I'm guilty of it too, but a study came out recently that it was a freeze-kill brood-based study that concluded there was no impact of hygienic behavior on the development of European foul brood in honeybee colonies. And so, it just made me, I read that and I thought, well, I wonder if they would have seen the same results if they'd used Harbo-selected hygienic colonies or UBeO-selected hygienic colonies. And maybe they would have seen the exact same thing, but I don't think it's a safe assumption that they would have. And so I just want folks to be careful about referring to hygienic behavior as if it's like one clean, neat thing, because it's so nuanced and so complex.
It's all about the definitions. As I start this conversation, the whole conversation can go to many, many different directions, depending only of the definition of hygiene. And people have the perception, the anthropological and perception that hygiene is something that we clean. We compare to ourselves. Like that's why people make the mistake to compare grooming with hygienic behavior, because grooming is some sort of hygiene, that people are removing things, just cleaning things. That means hygiene for us. But in the bee world, those words have different meanings, and people get confused.
Yeah, absolutely. Yeah, it's like saying sick people shouldn't go to work. Well, what do you mean by sick?
What do you mean by sick? What do you mean by work?
Do you mean they sneezed once, or do you mean they're, they've had COVID and they're highly infectious, and or is work at home? It's all about the definitions, like you say. So I just want to kind of bring awareness about that and make sure that we're all being clear when we say things, and we're meaning what we say and saying what we mean.
Fantastic. See, that's a salad of words right there.
Yeah, exactly. And I think the way to start resolving and start understanding these nuances is by going deeper into the biological mechanisms behind hygienic behavior. So understanding what is happening at the physiological level of the honeybee. And so I just wanted to kind of go over a little bit about what we know in terms of the biological mechanisms of hygiene. So there have been, dozens of studies on this. I can't list them all here, but we know that hygienic workers have better olfactory sensitivity. So this was a 2003 study based on freeze-kill brood. We talked about it earlier, that the bees that are performing hygienic, specifically uncapping, are more sensitive to certain smells. We know from a study that I did, and I'm in the process of publishing right now, that hygienic bees selected based on UBeO or colonies that scored high on UBeO have better long-term memory of unhealthy brood odors. And we know that genes associated with hygiene, or colonies that score well on hygienic tests like SMR, have genes that are associated with things like neural sensitivity, perception, and olfaction. So this paper is actually a review of, probably 15, 20 different other papers related to these. And we see consistency in kind of the characteristics of hygienic behavior. And so what that tells us is that smell, what each of these tells us is that smell and perception detection of these odors is really important. And so, of course, that means that the antennae are really, really important to hygienic behavior. And so the thing that I'm really interested in looking at in the antenna is odorant binding proteins. So odorant binding proteins bind and transport molecules that essentially come into contact with the antenna to receptors inside the antenna. And in honeybees, there are 21 of these odorant binding proteins. And there's been some really neat work looking at freeze-kill brood selected bees, where they found there was actually higher expression of two of these odorant binding proteins, number 16 and 18, in these freeze-kill brood selected bees. So the bees that are responding to freeze-kill brood more efficiently have a higher expression of these two odorant binding proteins. And then another study showed, well, these two odorant binding proteins actually can bind those necromones that are like oleic acid that are released from the dead bees. So that makes a lot of sense. Mm-hmm. So we said, hey, I wonder if this would be the same with UBeO. And so we thought we would try looking at the expression of odorant binding proteins from the high UBeO colonies. And we expected to see these same results where, we'd have higher odorant binding 16 and 18, odorant binding protein 16 and 18. We did a preliminary study with just a few colonies, and we found the opposite of what we expected, that the high UBeO colonies actually had lower odorant binding protein 16. And, at first I was really surprised, but then we noticed there was higher expression of a different odorant binding protein, not 16 or 18. And so I think this, lends even more support to the general message I'm trying to say that, UBeO is not the same as freeze-kill brood. They're similar, there's overlap, but when you get down into the biology, the physiology of what's actually going on in the honeybee body, there are different things happening. And I think that makes a lot of sense in terms of what we see. So bees that are selected based on removal of that dead bee odor have higher expression of proteins that can bind and essentially recognize dead bee odors. And so now we're running another larger study on this to see if we see this in a larger population, but also if the colonies that have higher UBeO and more of this other odorant binding protein also bind, if those proteins actually bind these UBeOs, which is what we expect. So it's really interesting to kind of tease apart the biology, the molecular genetic biology of what's going on in here, and differentiating these different types of hygienic bees.
So I'm assuming the X is a secret for now.
For now, yeah. Before I get it out there, I would like to confirm it on a larger number of colonies. But I am, collecting this week. Next week, I'm collecting samples for this study, and then I'll ship them off to my collaborator, Leonard Foster, in Canada, and I can't wait to see what he finds.
Yeah, curious. That's fun. That's fun research. Yes.
It is. And so, kind of just summing up my general message, all of these UBeO, freeze-kill brood, Harbo are methods for selecting hygienic bees, but they're not the same as each other. There are trade-offs between the different selection methods. As much as I'd love to tell you UBeO is the best, there are several great selection methods, and there are trade-offs for all of them. It really depends on what your goals are, what your resources are, in terms of what works best for you. Different selection methods can lead to different outcomes. I think that's important. Again, I think it's overlooked. And we have to be careful with generalizations about hygiene because it's really complex. It's really nuanced. It has a lot of potential, but there's so much left to learn. And like I said earlier, the deeper I dive into it, the more questions I have. And there's just a lot of work left to be done to really understand this behavior.
Yeah, so generalizations there from the beekeepers and also from the researchers. So
Exactly.
just to be clear, all of us are guilty of those generalizations sometimes.
Absolutely, and myself included. I'm definitely not immune.
Me too.
But it's something I've caught myself on, and I'm working on. So it's been on my mind a lot lately.
Cool. Oh, thank you, Kaira. Let me see if somebody have questions. Kyle just said that he had some question, but we already answered, so he's thanking you, Kaira.
Oh, my pleasure.
If anybody else is out there and have questions, now is the time. Otherwise, I'm gonna wrap this up with Kaira and let her continue her journey to find out new stuff and bring it to us later on.
Yeah, I think one of the other complications I'll say with hygiene is we're seeing that the environment can have so many impacts on the test outcomes. And so I think there's also a lot of work to do in unraveling the effects of season and nectar flow. And, we've already started to tackle some of those questions about like the effects of smoker use. So we see smoker can kind of dull the senses of bees a bit.
Yeah.
And we're working on trying to understand how other factors impact hygiene. It's just, I think measuring behavior is always difficult in any species. But when you have a social insect with tens of thousands of individuals and multiple mating, and there's just so many environmental factors that can have an impact. It's a lot to untangle, but fun to do, I'll say.
Kaira, question for you. Margarita is asking you, can you clarify why hives tested at different times, month, same queen, have different results?
Yeah, well, that's kind of exactly what I was just saying. We're working on figuring that out. I think that nectar flow, we've done an experiment that showed nectar flow can have a big impact. So I think that's a big part of it. I also think that the colony has different priorities at different times of the year. So if you think about the potential threat of a varroa in April compared, and this is Northern Hemisphere I'm talking about, but, a varroa in April here in spring is much more of a threat to the colony than a varroa in August or September, because the varroa in April has the potential to become many varroa over the next couple of months, whereas the varroa in August is essentially going to lose her access to brood pretty quickly, and so maybe the colony in August is focused more on upping their brood population numbers, gathering any last nectar sources they can, and so they're more workers focused on foraging or other duties than on hygiene. And so I think we have a lot of work to do to figure out what environmental impacts affect hygiene and how they affect scores. But I think that those are two primary reasons that you would see differences. So the nectar flow and then the colony priorities.
You have data on colony size? We do. The colony size could have a big impact on that.
We do have data on that, and we actually just published a paper on it. And at the colony level, we didn't see any effect on colony size. So we did, Phoebe Snyder did this work for her master's, and she showed that as, you go from like 3 bees to a few hundred bees to a few thousand bees, there's a big impact on the amount of hygienic behavior that's done. But when we compare like a nuke, a 5-frame nuke to a double deep with 2 supers, there's really no effect of size. So once you hit a certain threshold of bees, there doesn't seem to be any impact on of size of the colony on the hygiene, which is good because that means you can kind of compare all of the colonies in your apiary at one time. So to further kind of answer Marguerite's question or address it, one thing I'll say that you can do is any test that you do kind of at the same time under the same environmental conditions, you can compare with high accuracy those colonies in those tests. When you start trying to compare, a colony that you tested in April to a colony you tested in August, you can't always guarantee that that's a reliable comparison.
Very interesting, Karen. All this work has been decade now, and it's just a lot of fun following you from the very beginning, and also being part of your journey, and a lot of fun to watch all of this. I feel blessed that I could be so close by and see this whole from very little details here and there.
It's been a lot of fun to do. It's a lot of work, and I feel that there's a lot of work left to be done, but luckily I'm up for it. I'm interested. I'm so curious to answer all the questions I have. And so funding willing, let's see if I can get some grants funded. And, I have a lot of research plans to get some of these questions answered, but it depends on which things get funded and that I can really devote my time to. So I look forward to answering the questions that folks have, and I'm always interested to hear what questions beekeepers have, because those are the ones I want to address first.
Cool. Yeah, I'm looking forward to see what's going to happen next on your career. Some big decisions will come. I'm sure about that.
Yeah, we'll
Company
see.
versus university, or you can't. I don't think you can keep both forever. So.
Yeah, we'll see. For now, it's working, and yeah, who knows? Maybe I'll need a CEO at some point so I can move into the research realm a little bit more completely. But for now, it's really fun, kind of exploring both worlds and doing what I can to get this tool out to beekeepers.
And by the way, you're doing a fantastic job by yourself, doing this whole thing with the help of a few people, and you did something that very few people are able to do. So congratulations on that.
Thanks. Well, I definitely don't claim responsibility. There's lots of brilliant hands and brilliant minds and hardworking hands that have been involved in this work over the last decade. So lots of people to thank and acknowledge in getting Optera and UBeeO where it is today. Definitely not doing it myself.
Last message to people at home, Kaira. If you have anything extra to say, now's the time. Otherwise, I'm going to wrap this up.
Yeah. I mean, consider hygiene if you haven't. And if you have, consider the type of hygiene that you're working with, because I think not all hygiene is created equal.
That's great. And if you want to get UBeeO, use the link in the description of this video and use the coupon code Inside the Hive TV for 15% off, because she is being very generous to the people of this channel. So thank you for that, Kaira. That's being very nice for that.
Absolutely. Optera loves Inside the Hive TV.
Thank you. And that being said, I want to thank you, Kaira, and you guys at home for watching Inside the Hive.tv, the show about bees. See you guys next week. Bye-bye.